I hope this works for me. My name is Oscar Hennig. I'm the operations director for Benchmark Genetics on the shrimp division. I'll go fast on this. At Benchmark, we have genetic division, advanced nutrition, and animal health. Now going into a bit more on my cup of tea, genetics. We are big in salmon, we're getting big in shrimp, and also in tilapia. Those are the three species that really show the power of aquaculture. As mentioned before in the presentations, salmon is everywhere, so is shrimp. So what products do we have on the salmon? The main product is egg, the ova. On shrimp, broodstock mainly, but some small amounts of pos larvae or PLs being sold in the US and also in Colombia. Tilapia, broodstock mainly, but some sex reverse fingerlings within the US, Canada, and Mexico as well. And also we provide genetic service for other species besides salmon, shrimp, and tilapia. And as in salmon, we believe that one size does not fit all, and we have different lines of breeders, different genetic lines for different environments. BMK Yield is our balance line. Protect is our resistant line for white spot, but also for EMS, as was mentioned before. And our BMK Low Sal is a fast-growing line designed for lower salinity. There's a tendency of moving Vannamei shrimp, it's a saltwater species, but moving into inland in more or lower salinity. Just a quick, to put in perspective, about 48%, close to half of all the Vannamei farmed in China is farmed in lower than 5 parts per thousand. So it's quite important market. Also in India, it's about the same percentage. So where are we? So besides our salmon in Norway, Iceland, and Chile, we have our tilapia operation, our breeding operations in the US, south of Miami. We have our nucleus breeding center in Colombia, in the city of Cartagena. We actually have 2 nucleus breeding centers. The main one is in Cartagena, but we have on the Pacific coast, on the city of Tumaco, we have our another breeding center as well. Our elite multiplier is in the US, in Florida, just about an hour south of Orlando, where we have a 700 meters deep well water that pumps out 32 parts per thousand pristine ocean water that we use in our facility. So this is biosecurity is second to none. And just start operation, we start selling breeders from our MC multiplication center in Thailand, in the city of Prachuap Khiri Khan. And we just sold our first batch last month. So this was, there was a rubber bank mark here somewhere, but I guess it cut it out. So anyhow, as you all see, close to 5 million metric tons. And over that, about 80% is Vannamei. And Vannamei is a native species to the Pacific coast of the Americas. So from South California to Northern Peru. And 70% of all Vannamei produced is produced in Asia. So the only country really in South America that can compete with the Asian countries is Ecuador, as mentioned before. So what we are doing, and that's the main focus of this presentation, is showing that what we learning from the salmon industry at Benchmark, what the technology that we developed, and what we brought into shrimp, tilapia, and other major species. I'll be focusing on shrimp due to the presentation here. So Benchmark Genetics Shrimp, on the genomic side, we started in 2016 with a parental assignment SNP ship. We move into 2018 with a SNP array done together with Neogen, a custom SNP array. And in 2019, we add additional SNPs to be able to do the whole genomic sequencing for VNMA. And what that allowed us, I mean, again, that was technology that was brought from the salmon industry into the shrimp industry. What before 2016, for the Colombia operation, every single shrimp had to be hand tattooed or tagged with elastomer tag with different colors, different positions, so we can read and know which family from the 200 families that you produce, which family those animals were from. But in order to do that, you had to wait until they were about a gram and a half, 2 grams. And that's about 60 days on the production cycle from spawning to reach that size. That means a 3-month cycle animal, you keep them the first 30 days. So let's say the first 30 days at a individual rearing environment. So that allowed and that add too much noise to the selection process. And with the parental assignment that we were able to start using in 2016, that is gone. So that restrictions that imposed to us is gone, and we're able to mix them at nuclei stage, which is the first stage, larval stage after hatching. And just to put in perspective, this is way more precise and less foolproof than actually tagging the shrimp. Tagging the shrimp, reading the tag of shrimp, allowed more errors to be made, my human errors to be made, than by doing this. And then I just have a quick example of the last batch that we end up having 5 animals after parental assignment that we couldn't find. We didn't understand where were they from, because they belong to a female A, but they belong to a male B that really never spawned. We inseminated that female with male B, but she didn't spawn. We scrape, we add the new sperm from male C, and at the end, that female had progenitor with the 2 males. 50 animals recuperated after the trial from male C, and then 9 from male B. So that shows the power of this technology and how precise it can be for that. So now the whole genomic sequence, it's helping us develop SPF. You heard this term over and over, specific pathogen-free shrimp. But we developed, SPR, specific pathogen-resistant shrimp, from SPF populations. What Ecuador has done, that selecting animals that were the survivors, yes, they are the survivors, they are doing well for Ecuador, but you cannot export that product. That product, as a breeder, is contaminated. It has disease on it. It's are able to resist or to tolerate that disease, but still a non-SPF shrimp. But by using genomic selection, you were able to select the best families, like George Chamberlain mentioned, that you send cohorts of the animals and you receive the data back. That way, you use that data to select the best families. But with the genomic selection, with the training population that you have done, you were able to select the best individuals from the best families. And that gives you a push, especially, as mentioned before, for white spot and other diseases that has really low heritability. Okay, one more technology that was brought in from the salmon industry into the shrimp industry is breed control. That was a software database that was developed for salmon by Benchmark that now is being used for the shrimp industry. So we have up and running the maturation module with over 5,000 data points for different KPIs for maturation performance. With that, we're able to track performance and compare over time per country, per client, per batch, per line, protocols, and one that is close to my heart, ablation compared to non-ablation. For the audience, if you don't really know, ablation is when you cut the eye of the female. They reproduce more often and produce a larger number of eggs and nuclei. So we are advocating with our clients for non-ablation. But again, using the Power BI reports, we're able to track those KPIs. We are in the process of developing a larvae module and a farm module as well. So to give the understanding about the power of this data system that we've been using is that we noticed early on, this is country A, let's call it country B, and we see the same batches that were sent to different countries with different outputs. So using the KPIs, we want to know, okay, what is the major difference? We've seen that the total output of nuclei per female per month is incredibly different, but where are the differences? So mating ratio, meaning how many females are mated per night, percentage-wise. And we didn't see that much differences between the two countries. And average nuclei per female per spawn, meaning how much does a female spawn in one time. That's where we saw the biggest difference in country A, way higher than country B. And being a company that have advanced nutrition in its portfolio, we were able to see that nutrition and also management was the main issue for these big differences, and able to help the client and help solve this problem. Again, non-ablation is close to my heart. I want to see all the shrimp farming being performed with non-ablation. And we are starting our site in Colombia and also our site in Florida and our site also in Thailand, all with non-ablation. All producers are with non-ablation. And again, all our lines, our shrimp genetic lines are selected for non-ablation performance. And again, market perception is changing. There is anecdotally that PL from non-ablated females perform better than PLs coming from ablated female. We've done many trials, and yes, the eggs are bigger. There are more nutrition profile, which all makes sense. And we are pushing this to our clients to go for non-ablation. So again, we use the maturation module of our data system to understand where is the main difference. We know that compare total nuclei per female per month, an ablated female has roughly about 23% higher production output. Now, breaking down to KPIs, we saw that again, nuclei per spawn were about the same. So 5% higher for ablated females, but they were about the same nuclei per spawn. But the main driving factor was mated females, meaning how often a female mature, it enlarged. So from 5 days, it goes to 7 days, more or less. And that decreases the total production per month, not the total production per spawn, but the total production per month. And with that, we're able to know and start selecting for females that do come up with no ablation, that do come up more often, working on the genetic side, but also working on the day-to-day practical side to push our clients to adopt non-ablation, is to offer more females, roughly 20% more females than male. Usually the sex ratio when you sell breeders, shrimp breeders, is 1-to-1. But we're offering 20% more females to compensate for now for this. And it has been quite well accepted. Now, just to close, our performance, it has been, that's one of the reasons why we have different lines of shrimp or breeders, genetic lines, is to produce and to give the farmer a stability of production. You saw on previous presentations how shrimp production fluctuates quite a lot. So that has been benchmark goal, is to produce a more stable, to give our clients and the clients of our clients, which are the farmers, a more stable and more predictable harvest. So we see here our balance line in Indonesia producing higher yield, kilos per hectare. But one thing is, when you look at the coefficient of variation for ADG, average daily gain, you see that is a lower lower percentage of coefficient of variation, so a more stable growth when compared to our competitors. And also the survival, the coefficient of variation for survival is lower with a higher survival, but more stable survival than our competitors. And with that, I thank you all very much for your attention, and any questions or anything, I'll be more than glad to answer. And then again, this is our site in Florida, again, surrounded by agricultural land, as biosecurity as it can be.